Happy New Year to everybody and good morning. This is Jin San Yu. I'm a CEO of Pharmapsin, a clinical stage public company, mainly focusing on next generation antibody therapeutics. Who we are. Pharmapsin was founded on September 3rd, 2008, by engagement from Novartis. And we successfully closed our Series A, which was co-led by Novartis and OrbiMed in the global economic crisis, which we are very proud of. We are mainly focusing on development of next generation antibody therapeutics. We are listed at KOSDAQ. As I mentioned, we are a clinical stage company. Our lead asset, called olinvestmab, is anti-VEGF2 neutralizing fully human IgG1 with best-in-class potential, and is currently in 2 phase 2 clinical studies. Originally isolated from Huphase display library, we developed as a fully human phage display antibody library with higher diversity and highest quality. 2 first-in-class preclinical assets that are being prepared for phase 1 in this year. One is PMC309, which is anti-VISTA neutralizing fully human IgG1, and the other one is PMC403, an anti-Tie2 antibody which activates the downstream signaling of Tie2. It's also fully human IgG. We have one subsidiary called Winkel Biopharm located in South San Francisco in the States, and the other subsidiary located in Brisbane, Australia, called Pharmapsin Australia. What we are looking for at Biotech Showcase, we are looking for opportunity to licensing out our olinvestmab and to license out or co-develop both PMC309 and PMC403, which are in IND enabling stage, and file IND probably Q2 of this year. We are also looking for investment from strategic investors, and we are always looking for research collaboration opportunity for all stage assets what we have. This is selected pipeline status of development. And as I mentioned, olinvestmab, with Merck together, we initiate 2 combo trial with Keytruda and olinvestmab for metastatic triple-negative breast cancer as second line and third line, and the other indication is recurrent GBM. We also have olinvestmab monotherapeutic for Avastin non-responding recurrent GBM trial in both United States and Australia. And PMC403, we are challenging 2 indications, one in age-related macular degeneration, diabetic retinopathy, and diabetic macular edema in ophthalmology area. And also, we are exploring oncology phase 1 trial. And for PMC309, which is anti-VISTA neutralizing antibody, of course, for oncology, and also probably Q2 of this year, IND filing. So, olinvestmab, which is VEGFR2 neutralizing fully human IgG1. That's a key clinical results and status what we have. The true competency of olinvestmab is truly clear safety profile compared to Avastin, Xarelto, Geltrap, or even small molecule like axitinib and lenvatinib and those things. So no hypertension, no GI perforation, no proteinuria, no intrableeding hemorrhage. Only we observe is grade 1 or grade 2 reversible capillary hemangioma on facial, neck, and chest area. We have shown our phase 2A recurrent GBM trial in Australia previously that it has delivered 25% disease control rate, maximum 60 months. Patient survive with high quality of life. Olinbasimab also provide edema relief, the cerebral edema relief in 42% patient of phase 2A, and tumor reduction for 25% patients. And due to the edema relief, we also reached 70% patient has steroid use reduction and survival up to 525 days. Olinbasimab for primary and recurrent GBM was orphan drug designation granted by US FDA and Korean MFDS. And 15 out of 36 patients were recruited for Avastin non-responding recurrent GBM monotherapy, which is currently running. And we recently recruited the first patient for phase 2 Keytruda combo study for second and third line metastatic triple negative patients. In more detail, the combo trial for recurrent GBM is Keytruda and olinbasimab. The total enrolled patient number was 9, and it was subdivided into olinbasimab dose was 12 mg per kg and 60 mg per kg. And as always, no DLT and grade 3 adverse effect was just one event, which is fatigue. And severe adverse effects is 6 events for 4 patients. And as I mentioned, the capillary hemangioma on the skin is grade 1 or 2 occurred from 6 patients. Efficacy, so 4 patients, which is 44%, had a stable disease as best response. Last patient, last visit was completed at 35 cycle, which is about 24 months. Merck has internal policy that they always stop Keytruda treatment after 35 cycle, regardless as a monotherapy or combo therapy. So this patient, of course, is stable disease condition, and the last one patient has been treated with olinbasimab for 4 cycles as compassionate use since Keytruda treatment was stopped, but PI and patient and caregiver are asking for compassionate use for olinbasimab, and we did that, and those studies still running. In case of metastatic triple-negative breast cancer, which is more exciting, 11 patients were recruited in total. 5 patients in 12 mg per kg olinbasimab cohort, and 6 patients for 60 mg per kg olinbasimab cohort. And again, in terms of safety, we have a clear safety profile. Efficacy-wise, in higher dose of olinbasimab, we have 50% ORR and 67% DCR. And one patient is staying over 33 cycles at the moment, and one patient which has CR and target lesion has been treated with olinbasimab from cycle 30 due to side effects from Keytruda. This patient has been treated with olinbasimab for 12 cycles, so which 7 cycles were compassionate use. So this is the waterfall plot. So you can see the tumor size change, which is quite promising in second line and third line metastatic triple-negative breast cancer. So you know, Keynote 109, which is Keytruda monotherapy for same target patient population, just delivered below 10% ORR. So because of that, Morgan Palmaption filed combo data for new IP and also opened the combination phase 2 trial. And good thing is we had the first patient enrolled, and the treatment was the infusion is completed, and patient's hepatic was clear and went home back. So there will be 35 cycles as usual in maximum. And when the patient status is good, we are willing to provide onvansertib compassionate use after 35 cycles is completed. We will also open additional sites in Korea and plan to file IND in Q1 of this year. So I'm going to shift the gear to two preclinical stage assets. One, PMC309, anti-VISTA antibody, and PMC43, which is tied to activating antibody. So VISTA is quite new in immune checkpoint blockade list. However, several antibodies are running phase 1 recently, and we are also preparing for IND this year. So highly expressed on myeloid-derived suppressor cell, MDSC, or myeloid cells, and to a lesser extent on T cells, including Tregs and tumor microenvironment. VISTA blockade also improves anti-tumor T cell responses reported in many different organizations. And combination therapy to inhibit VISTA and other negative checkpoint regulators on multiple cell types will cover broad patient population, including patients resistant to anti-CTLA-4 antibody or also anti-PD-1 or PD-L1 antibody treatments. Interestingly, also PMC309 regulate T cell activity. PMC309 showed a highly selective binding to VISTA in various experimental settings, including in a low pH environment. PMC309 shows mild ADCC effect, while it shows strong ADPC effect, suggesting there is some T cell activity, but also macrophages. T cell activation is induced upon PMC309 binding to monocytes. As you can see in our ex vivo studies, in the presence of PMC309, the T cell activity increased in dose-dependent manner. And regulatory T cell depression also decorrelated with dosing of PMC309. We also identified that PMC309 shows strong monocyte activation than anti-PD-1 antibody or anti-PD-L1 antibody or anti-CTLA-4 antibody. So CD14 positive human monocytes and CD3 positive T cells were isolated from human PBMC and were cultured in a plate coated with 2.5 microgram per ml of anti-CD3 antibody. The culture was incubated with LPS to initiate monocyte activation and humanize G1, PMC309, anti-PD-1 antibody, anti-PD-L1 antibody, and anti-CTLA-4 antibody was added to the culture. The upregulation of macrophage population and macrophage activation were measured after 24 hours. And as you can see, that in the presence of higher dose of PMC309 compared to CTLA-4, PD-1, PD-L1 antibody, the activity of M1 macrophage enhanced much higher. And also, if you look at the TNF-alpha secretion, it has high activity compared to anti-CTLA-4, PD-1, or PD-L1 antibody. We also has confirmed that PMC309 shows anti-tumor activity in vivo models. Firstly, PBMC humanized human TNBC animal model system. As you can see, the dose-dependent manner of anti-tumor effect, but also synergistic effect combined with PMC09 with anti-PD-1 antibody. And as you can see on the right panel, in human VISTA knock-in mouse model, also reconfirm that PMC09 has tumor effect in dose-dependent manner, but combination with anti-PD-1 has better performance, as you can see. And PMC09 induced an increase in the tumor, in the number of tumor infiltrating lymphocytes, especially CD8 positive T cells. So we are very excited to file our IND for PMC09 in Q2 of this year, and we'll see how it goes and how we differentiate from other VISTA antibody competitors. PMC43, that's a novel anti-Type2, highly binding, fully humanized H1, and not antagonizing Type2 biology, but opposite. It's agonizing the downstream of Type2 biology and somehow stabilize and normalize pathological vasculature systems. And it can apply various indications, not only oncology or ophthalmology, but other areas of disease condition which related to dysfunctional, disorganized, leaky vasculature systems. Interestingly, angiopoietin-1 and angiopoietin-2 bind to Type2. And so far, angiopoietin-1 activate Type2 and stabilize vasculature system, whereas angiopoietin-2 has a kind of opposite biology. Also, when we are aged, somehow the vasculature systems are also aged and dysfunctional. And there is a study showing that when the vasculature system and we in our body aged, the Type2 expression level decline on the vasculature system, and somehow that is some relationship with dysfunctional vasculature system too. And interestingly, Type2 also plays pivotal role for choroidal blood flow. However, no significant biology in retinal blood flow. So we first tested PMC43 in tumor models in EGFR variant positive GBM mouse model system, and somehow PMC43 organize and normalize the vasculature system in mouse brain related to GBM tumor microenvironment. And also, we confirmed that in CT26 colon model system that somehow it's not dramatic, but there's some anti-tumor effect, probably linked to the reorganized vasculature system, provide efficient delivery of innate immune cells, so that the immune cells can enrich on tumor microenvironment and deliver some anti-tumor effect. We also checked in CMV mouse model system. As you can see, this is kind of apple-to-apple comparison study with EYLEA and PMC43 to repair the leakiness of the damaged vasculature system in the eye. And Also, in mouse model, the ERG test is comparable to ILEA and PH43. And because of that, we moved to CMV monkey model system, and also reconfirm that both ILEA and PH43 can repair the retinal leakage, similar efficiency, and also managing the retinal thickness compared to ILEA. PH43 has well, slightly better, but I would say it's a similar. But the optic nerve response, the ERG test, PM43 is better than ILEA. And due to the limited number of monkeys, we designed to reconfirm this higher number of monkeys for these studies. And when we look at the gene expression analysis, angiogenesis and vessel normalization is similar, and anti-inflammation effect are slightly better than ILEA. So also like PMC serial 9, PMC43 is heading to IND filing in Q2 of this year. We are heading to IND filing, and we initiated our whole IND package study in Q2 last year. And in oncology, so since it is two studies, one in ophthalmology and the other one is oncology, we are also heading to oncology GLP PK tox package, and will be completed in Q1 this year. So we are open for any constructive and productive collaboration and business development. So if you have any questions, do not hesitate to contact me. My email address is junsanet.u@pharmapsin.com. Thank you.